Hello and welcome to another in a series of phytosanitary irradiation webinars. My name is Paul Wynne. I'm the host of today's webinar and chairman of the International Irradiation Association. The Phytosanitary Irradiation Platform, otherwise known as PsIP, was established a few years ago by irradiation experts who wanted to establish a forum for information exchange, networking and collaboration around the world. There's no cost to being part of our community, so if you're not already a member of PsIP, please visit the website psipglobal.org and sign up. PsIP is a focused community and is part of the an IIA initiative. The journey to forming PsIP commenced in 2018 when IIA took a lead in organising a phytosanitary meeting in Bangkok. This followed a very successful series of annual meetings organised by Chapman University in the US. In 2022, PsIP returned to Bangkok to host a phytosanitary radiation forum as part of the International Meeting on Radiation Processing. Before we begin, a few housekeeping points. The webinar is being recorded and it will be available via the PsIP website. The chat function has been disabled, but you will be able to submit questions via the Q&A button. After the presentation, there will be an opportunity for questions. The presentation will last about 35 minutes and will then allow 10 to 15 minutes for questions. Today's presentation, Comprehensive Generic Doses for Phytosanitary Irradiation, is being given by Dr. Peter Follett, who is a research entomologist at USDA, the Agricultural Research Service based in Hawaii. Peter has been an active researcher in applied entomology for more than 30 years and is internationally recognized as an expert in post-harvest entomology and quarantine treatment development. He's published over 260 articles, including 190 in peer-reviewed journals. And in 2017, he co-authored a book, Irradiation for Quality Improvement, Microbial Safety and Phytosanitation of Fresh Produce. Peter, welcome. May I invite you to give your presentation? Thank you.
Thanks, Paul, for the introduction. Okay, hi, this is Peter Follett speaking to you from Hawaii. As Paul said, I'm an entomologist and researcher with the USDA Agricultural Research Service located in Hilo, Hawaii. For many years, I've been developing phytosanitary irradiation treatments to control insect pests and fruit so that we can safely export fruit from Hawaii to the US mainland. I work closely with both farmers and with the two commercial irradiation facilities here to address problems and look for new export opportunities. Worldwide, of course, the use of irradiation for phytosanitary purposes has been rapidly expanding. Generic doses have been the key to this expansion. Today, I will discuss what I think is an important next step, the establishment of comprehensive generic doses. Food irradiation is a technology that improves safety and extends the shelf life of foods by reducing, deactivating, eliminating microorganisms, deactivating or eliminating microorganisms and insects. It is also a means to control quarantine insects in fruits and vegetables, thus allowing the fruit to be exported safely without pests. This presentation focuses on the quarantine uses of irradiation. I'll first talk about what quarantine pests are and how to control them, then dive deeper into different methods of control and the advantages of using irradiation, and how generic irradiation doses have accelerated the process of gaining market access. Then I'll present a short course in entomology and the insect classification system to help explain how we develop generic treatments for various insect groups. And finally, I'll propose a slightly different and more comprehensive system of generic doses, which will lower the required dose in many cases while avoiding gaps in coverage. As we all know, globalization has resulted in greater trade and transport of agricultural commodities. This in turn has facilitated the introduction and spread of invasive pests to new areas. This picture shows the surface traffic of ships around the Pacific over the course of a year. Hawaii is a group of islands in the middle of the Pacific Ocean and a common transoceanic stopover. Hawaii has a mild climate, which is friendly to new invasive insects. In fact, about 1/3 of Hawaii's insect fauna came from somewhere else because of people's comings and goings. Here I show several significant fruit pests that landed in Hawaii, including the mango seed weevil and melon fly from Asia, the lychee fruit moth from Australia, and the white peach scale originally from South America. Many of our pests are not found on the US mainland, so we need to apply a quarantine treatment to control the pests before exporting fruit. The same is true for many countries that trade in fresh fruits and vegetables. To illustrate, the melon fly, shown here, is one of Hawaii's most important economic and quarantine pests of fruit. It's a regulated pest because it is established in Hawaii and not established anywhere in the continental United States, which is our main export market. When the larval stage matures and drops from the fruit to the ground to pupate, so it is the larval stage in the fruit that we are trying to control with a quarantine treatment. Thus, as is the case for all taffruted fruit flies, we're trying to control that larval stage. Hawaii also has the Mediterranean fruit fly, the Oriental fruit fly, solanaceous fruit fly, and olive fly. We must demonstrate that any quarantine treatment can control the eggs and larvae in the fruit before we can get an export protocol for market access. The need for quarantine treatments is the same all over the world. One country has pests that the other country doesn't want. So the pests must be controlled before trade can go forward. The main options for quarantine treatments are fumigation, cold, heat, or irradiation. Fumigation is far and away the most common type of treatment, with methyl bromide or phosphine or sulfuryl fluoride or ethyl formate. Fumigates are easy to apply to large volumes of fruit, but may cause phytotoxicity in some cases and leave residues. Cold treatment is practical for some fruits that can tolerate temperatures near freezing, but takes a long time to apply, like 2 to 3 weeks. Heat treatment is an option for certain fruits, but usually causes some loss in quality. All these treatment types are developed one at a time for individual pests on a specific fruit, and research and approval of treatment protocols can take years. Irradiation is different due to the availability of generic treatments, which are approved for all insects on all commodities. So, irradiation can be used immediately, regardless of the commodity and pest, without further research. Irradiation is broadly effective against insects and mites and other types of quarantine pests, and generally does not cause any loss in fruit quality. For some fruits, shelf life may be extended. It is cost competitive with other types of treatment and can be applied quickly. Products can be treated in their final packaging, unlike other treatments, and irradiation can be applied at any temperature. Due to the massive damage to the insect's DNA caused by irradiation, resistance should not be a problem. A big advantage is the availability of generic treatments. Irradiation does not cause high mortality in the short term, so live but non-viable insects may be found after export. This requires that treatment development be extra rigorous, and 100% mortality with high confidence must be demonstrated via large-scale testing. So what exactly is a generic dose? A generic dose is a single irradiation dose for a broad category of pests. The first generic dose was approved against all fruit flies in the family Tephrididae, which includes Mediterranean fruit fly, Oriental fruit fly, Queensland fruit fly, apple maggot, European cherry fruit fly, and many other significant fruit pests. The rationale is that related insects have similar radiation tolerance, so information on a few species can be extrapolated to the whole family. Before a generic dose is proposed, there should be radiation tolerance data for the major economic and quarantine pests in the group, and many of the studies should include large-scale tests that confirm a dose with high confidence. There will often also be several smaller-scale studies done with other species that support the dose, and no radically different results with a species in the group. The highest dose required to control the most tolerant species becomes the minimum generic dose for the group. Quarantine pests may be inside the fruit or outside on the surface, or they may not be pests at all and just hitchhiking. For control with irradiation, we may be aiming for the larval stage of the pest in the fruit, in which case the desired response might be to prevent adult emergence, or we might be targeting the adult stage on the outside of the fruit, in which case the desired response of the treatment would be to sterilize the adult. In both cases, we are trying to prevent reproduction so that the pest cannot establish if it is accidentally exported to the area without the pest. Irradiation may also be applied as an insurance treatment to an unregulated commodity to prevent problems if random pests are found unexpectedly during inspection at the destination. In the US, irradiation of fresh horticultural commodities is approved up to 1,000 grays, 1 kilogray. There are two major generic treatments in use today, 150 grays for tephritid fruit flies and 400 grays for all insects, except pupae and adults of Lepidoptera, which may require higher doses. The 1,000 gray upper limit is significant because when irradiation is applied commercially to aggregated product, like a pallet of boxes of fruit, the maximum dose may reach 2 times the required minimum dose or more, meaning that when applying a dose of 400 grays, some part of the product may receive 800 grays or more. All countries exporting fruit to the US must adopt these doses and other regulations. Australia and New Zealand, the FSANZ at the top here, are using the same generic doses with some additions. In addition to the generic treatments, specific doses ranging from about 60 grays to 300 grays have been approved for 24 individual quarantine pests. These treatments can be found in the USDA treatment manual online, and many of the specific irradiation doses found there are for pests in Hawaii to facilitate exports to the US mainland. Since these generic doses were published in 2006, many countries have requested market access for various fruits. Exportation of mangoes was a common request from many countries early on, and we can see how the generic treatments played an important role in facilitating approvals. In this figure, the y-axis shows the number of quarantine pests, and the x-axis shows the distance from the United States. The farther the countries are from the US, the higher the number of quarantine pests. For example, 4 high and medium risk pests of concern were identified for Jamaica, the closest country, and 20 pests were identified for India, the farthest country from the US. It would be impractical to develop specific irradiation treatments for 20 pests if you were India, but the availability of the 400 gray catch-all generic dose meant mangoes could be exported immediately without further research. With the availability of generic doses, the number of countries exporting fruit to the US has steadily increased, especially using the 400 gray all insects dose. The 150 gray generic dose is used for fruits where fruit flies are the primary pest of concern, and other pests are either easily removed from the surface of the fruit or are rare. Mexico is the biggest exporter of fruit to the US using irradiation, and guava is the fruit with the highest volume. The 400 gray dose was chosen as a safe dose for all insects after careful review of the literature at the time. It was set intentionally high so that it could control even the most radiation-tolerant insect species. Most insects could be controlled at a dose below 400 grays, however, and there are many advantages to lowering the required dose. For a commercial irradiation facility, applying a lower dose will reduce cost and increase throughput due to shorter treatment time. For fruits that may be sensitive to irradiation, minimizing the dose may be beneficial. So how can we lower the dose? Developing more generic doses that cover other broader groups of insects would be a good start. Let's consider a couple examples where either a lower dose or a new generic treatment might be helpful. First, fruit quality. Apples are generally very tolerant of irradiation. The 5 cultivars from New Zealand shown in this figure all showed limited internal browning up to 600 grays. But at 800 grays, one cultivar, Royal Gala, had a big jump in internal browning, which would be unacceptable commercially. If the requirement was for irradiation at 400 grays, some fruit on the palate might experience doses as high as 800 grays, illustrated by the purple bar here, and suffer quality loss. Fortunately, the main pest of concern for export markets in New Zealand is the apple leaf curl midge, which can be controlled at a dose closer to 150 grays, which might result in a maximum dose to the product up to about 300 grays, well within the tolerance for Royal Gala and the other cultivars. The next case is for a new generic dose. For years, sweet potatoes were exported from Hawaii to the US mainland using a dose of 150 grays to control 3 quarantine pests, including 2 species of weevils, the sweet potato weevil and the West Indian sweet potato weevil. This changed when a 3rd weevil, the rough sweet potato weevil shown here, was discovered in the islands. Now sweet potatoes had to be irradiated using the 400 gray dose, which was costly. We conducted research showing that this weevil was very susceptible to irradiation. It could be sterilized at 50 grays, but the weevil proved to be difficult to raise in the lab, so it would be hard to perform the large-scale test required to confirm a dose and propose a treatment. Many crops are attacked by different species of weevils, and this is a group that is being targeted for a new generic dose. The family is called Curculionidae, and data for about 15 species suggest a dose of 175 grays would be sufficient for the group. If a generic dose is approved for the Curculionidae weevils, Hawaii would be able to lower the required dose from 400 grays to 175 grays for exported sweet potatoes, which would be a big advantage, of course. So the USDA has approved 24 specific irradiation doses for different quarantine pests and 3 generic doses. The fruit flies, family Tephrididae, the leaf roller moths, family Tortricidae, and for all insects. A dose for the weevils, family Curculionidae, is pending. The IPPC recommended recommends irradiation and other quarantine treatments in the ISPM 28. So far, there are 20 specific doses recommended and 3 generic doses. For fruit flies, family Tephrididae. For leaf roller moths, family Tortusidae, like the USDA. And a lower 70 gray generic dose for the genus of fruit flies within the Tephrididae called Anastrapha, which includes Mexican fruit fly and South American fruit fly. Before we talk about generic doses more, it's useful to understand the insect classification system. We all recognize insects. They have 3 body parts or body regions: the head, the thorax, and the abdomen, a pair of antennae, 6 legs, and a pair of compound eyes. Insects generally are classified into broad groups based on their body plan, type of mouth parts, and wings. Insects have either incomplete or complete metamorphosis as they grow and develop. Within incomplete metamorphosis, the youngsters look like the adults, only smaller. This is a stink bug in this picture, but cockroaches would be another good example. With complete metamorphosis, there are 4 distinct stages: the egg, larva, pupa, and adult stages. Beetles, flies, and butterflies and moths all have this type, and the larvae and the adults of these insects often live very different lives in different places. We classify insects using the system of binomial nomenclature introduced by the Swedish botanist Carl Linnaeus in 1753. In this system, all organisms have 2 names belonging to a genus and a species. But the system starts much higher up up in broader classifications shown here. Humans, for example, are Homo sapiens, genus and species. We are related to other species up the ladder as primates and higher up as mammals. The system is similar in insects. On the right is the full classification for the Mediterranean fruit fly. The species name in red is Ceratitis capitata. It's in the family Tephrididae. As mentioned, this is an important family with many of our most important quarantine pests. The family Tephrididae is in the order Diptera, which includes all flies. Diptera means 2 wings, so all flies have 2 wings instead of 4 wings like many other insects. The first generic dose was developed for fruit flies in the family Tephrididae, and the reason was that this group had some of our most important quarantine pests. But also, we tend to recognize insects at the family level. For example, ladybugs are in the family Coccinellidae. Honeybees are in the family Apidae. Ants are in the family Firmicidae. Stink bugs are in the family Pentatomidae, shown here, etc. So it is natural to focus on developing generic doses at this level of classification, at the family level. The problem is that there are many families, many families of insects, hundreds of families. Quarantine pests are mainly found on a few important and well-known families, but some quarantine pests come from less well-known families. Let's first take a look at a couple of the generic treatments that have been developed at the family level. As mentioned, the family Tephrididae, the fruit flies, contains many of the important quarantine pests of fruit. Development of the generic dose for fruit flies of 150 grays set the trend for family-level generic treatments. The dose was set based on information from about 17 species, including medfly, several species of Ragelitas fruit flies like apple maggot and blueberry maggot, and several species of Bactrocera like oriental fruit fly and Queensland fruit fly, and several Anastrapha species like Mexican fruit fly. Another important group of quarantine pests is the leaf roller moths in the family Tortricidae. The dose was set at 290 grays by USDA based on data from 12 species, including codling moth, European grapevine moth, and light brown apple moth. This dose is restricted to eggs and larvae of the tortricids. Moths are generally tolerant of irradiation, so some species of moths are the most difficult insects to control with irradiation, and the pupal stage may require a higher dose in some cases. As noted, the IPPC set the dose for tortricid moths at 250 grays, slightly lower. Another important group of quarantine, as mentioned, the generic dose has also been proposed for the weevils, which is in the family Curculionidae. The dose is based on information for about 15 species of weevils. The sweet potato weevil and the plum curculio are good examples of quarantine pests in this group. And for mealybugs, which are in the family Pseudococcidae. Note that mealybugs are in the order Hemiptera, which also contains many other surface pests that may be regulated. So these are the most important families of quarantine pests. The fruit flies in the family Tephrididae, leaf roller moths in the family Tortricidae, weevils in the family Curculionidae, and mealybugs in the family Pseudococcidae. And we have developed or are developing generic doses for these families that will allow phytosanitary treatment at doses below the 400 gray generic dose. But there are many other families of quarantine interest. And if we continue to develop generic doses at the family level, we will be at this forever. So I propose we assign generic doses to insects at the next level up, the order level, rather than at the family level. I think we have enough data across all the important quarantine pest groups to support this idea. This will help avoid gaps in coverage while lowering the required dose wherever possible. Let's consider the order Hemiptera, which I just mentioned, where the mealybugs are found. In addition to mealybugs, there are also several other surface pests that are commonly found on certain fruits, including scale insects, psyllids, and whiteflies. These all belong to different families, but are similar in radiation tolerance to mealybugs. So why not just set a generic dose for all Hemiptera to cover all these important pests? And I think we can propose this for other orders as well. This graph shows the relative radiation tolerance among insects at the order level. You can see that moths are highly tolerant, whereas flies and beetles have relatively low tolerance. And many of the surface pests, like scales and mealybugs and thrips, are intermediate. The important point is that we have data for all the major quarantine pests at this point, and many additional species, and effective doses appear to be consistent across species at the order level. The conclusion is that we can characterize radiation tolerance in insects at the order level, and the application of phytosanitary irradiation would be improved by using these conservative doses. So instead of the family level as we saw before, we would assign doses at the next level up, the order level, which would include all the families in the order. In this case, all the families of beetles, in addition to the Curculionid beetles on the left, and all the families of flies, in addition to the Tephridid fruit flies on the right. The comprehensive generic doses at the insect order level would look like this. The 400 gray generic dose would only be used in the case where the pupal stage of a moth, which is the order Lepidoptera, is associated with the fruit of interest, which is not really that common. Therefore, for most fruits, a dose lower than 400 gray could be used, either 250 grays or 150 grays, depending on the mix of quarantine pests. This system is simple from a regulatory perspective and flexible so that its decision which dose to use could be made after routine inspection of the shipment at the irradiation facility. So to summarize, generic doses are the key to current successes and the wider adoption of phytosanitary irradiation. The 150 gray and 400 gray generic doses provide good coverage for most quarantine pests, but the 400 gray dose is overkill for most insects, and additional generic doses for other important groups or revision of the concept using order level doses will allow the use of lower doses, which will result in lower cost and increased capacity for commercial irradiation facilities and minimize any fruit quality problems. Several other obstacles to wider use of phytosanitary irradiation are shown here, and we could easily talk about these for hours, but I'll just bring them up. First, the labeling requirement is a hangover from the decision in the early days to call irradiation an additive rather than a process. Labeling may sound to some like a warning. This requirement should be eliminated. Many countries still have not approved phytosanitary uses of irradiation. For example, the US's main trading partners, Canada, the EU, Japan, South Korea, still do not have regulations governing phytosanitary uses of irradiation, and this has stymied interest in the US for export. And finally, the 1 kilogray limit was arbitrarily chosen 40 or 50 years ago and serves no purpose. This limit should be raised or eliminated, which would allow large-scale multi-purpose facility the ability to irradiate more efficiently. Anyway, that's it for now. Making these changes would clear the way for wider uses of phytosanitary irradiation, bringing us all to closer to a state of complete happiness and utopia. Thank you for listening.
Peter, thank you. That's extremely comprehensive, very fascinating. And just to remind everyone, there's a chat function through the Q&A. So if you've got any questions, please type them in, and they'll pop up, and we can ask them of Peter. I mean, you mentioned we're just seeing if there's any questions from the floor. You mentioned 2006 was the beginning of this. If you stay at the family level, this is going to take an awfully long time. How far down that journey do you think we are at this stage in terms of at the order level? Is it just, have we made that much progress so far? And are there any
we've made a ton of progress in the last 20 years, I'd say. And as I mentioned, I think we've got some information, in some cases really good information, for most of the important quarantine pests found worldwide. So this is why I'm proposing to sort of knock it up a level to this order level by developing or approving doses at the generic level. Basically, we just get much better coverage. We won't run into insects showing up that cause use of this high 400 gray treatment. So it comes down to regulatory agencies. So people like me do the research and propose changes, and then the regulators consider our suggestions and decide whether to go through the process of changing the regulations, which can take time. So again, I've been talking about this for a number of years, and there's been no movement, but I haven't really proposed it to a larger group like this. So we'll see if there's any movement or any interest.
And I suppose a connected question would be, how do we build that consensus? How do we get that agreement? There is a question here from Christine, which says, where are you in the process of adopting the new guidelines that you're suggesting? And that's a connected question, really, because do you see regional variations in this? And you mentioned the USDA, and we've got the ISPM, the IPPC regulations. How do we bring that together, and what's the process? What are the sort of timelines to achieving this consensus, and what role can this group and how can we assist in that process?
Well, that's a great question, Christine. Thanks. The USDA was the idea of generic doses has been around for 30 years. The USDA was the first one to implement 2 generic doses. Since then, Australia has adopted similar doses. New Zealand actually has been very progressive in adopting a full slate of generic doses for different groups. So really, it comes down to having the right people in position of making the change, I think. People with an open mind. So I think the best way is to get one country to make the change, and then hopefully the others topple and agree with the change and make changes themselves. So I'm not sure what the absolute best way to do it is. Educating people is the best start. A talk like this, I think, in front of an international audience, of course, helps a lot. And certainly the IIA's support and these sort of initiatives would be super helpful.
And the people representing different communities with it globally, I imagine, because it does seem that there is quite a difference between the direction of travel in Asia Pacific and the Americas, in a sense, and Hawaii, of course. You're straddling both of those. You're on that almost like the international date line. You're there with a foot in both of those camps, I suppose.
Yeah. So again, I'd mentioned, the U.S. is the biggest importer of fresh produce using irradiation, and Australia is the biggest exporter of irradiated produce. And Australia seems to be growing leaps and bounds. They're really exploring the export space using irradiation. The U.S., I think, is doing great too. But so many countries haven't approved phytosanitary irradiation yet that the U.S., as an example, a great proponent of irradiation, food irradiation in general, and phytosanitary uses of irradiation, is not interested in export because our main export partners have not approved phytosanitary irradiation. So I think that's a big hurdle for us as a country, and I'm sure other countries have run into this also. They're big trading partners, be it the EU or other countries, don't really have regulations in place to accept irradiation for phytosanitary purposes. So there are a few hurdles to clear still.
And do you see any a particular pest groups that would offer the best avenue for progress here? Are there particular groups of pests that are easiest to apply generic doses, comprehensive generic doses to?
This is I think is where regulators get kind of hung up. If you're going to propose a dose for a group, they want to see a bunch of species that have been tested, and a number of those species tested at a high level with large-scale testing and so forth. So it's ironclad dose for the group. So the 4 groups I've mentioned are sort of the biggest and most important groups, and we have the most information for those groups. But there are a lot of other groups. Mites are an important group that have been understudied. We recently completed a study with a slug, which is a problem on exported produce from Hawaii. There are relatively few studies with gastropods. So there are many other groups out there that can be studied. I think people are continuing to study. The IAEA has initiatives to develop further generic doses. I've proposed, additional generic doses for ants and for snails and slugs and things like that. But again, for many people, it comes down to a numbers game. Do you have enough numbers? To me, more important is, have you considered and do you have data for the most important and commonly intercepted quarantine pests? That's my view is a little bit different maybe than a regulatory person's.
Thank you. And if you can assist with this one, is there a possibility of pests being vectors for other plant pathogens, a major stumbling block? What do you make of that?
I would say I don't consider it a stumbling block at all. With the generic doses we have now, we have generic doses for several groups, but we also have specific doses for individual pests. And those doses tend to be lower than the generic doses. So I think the regulations are always full of exceptions. And if there are exceptions that we need to write into the rules, we just write the exceptions in. If Asian citrus psyllid is transmitting citrus greening disease, we can make an exception, or we can study it more fully and come up maybe with a more accurate dose for that situation. Interestingly, the snail I just talked to you about, it's one of several snails here in Hawaii that vectors a nematode that causes meningitis in humans, called a disease called rat lungworm disease. We showed first that the 150 gray and 400 gray dose controlled the slug, prevented reproduction in the slug. And then later, using rat models, showed that it also controls reproduction in the nematode, so that we could stop any transmission of human disease in exported produce, where whatever the chain of events happened where humans might be infected with this nematode. So those types of studies can be done. I don't think it's would stop progress on generic doses and so forth. No.
And a question from Chip asking about the 1 kilogram upper limit dose, and saying, is the FDA working on this, or has anyone recommended it? What's the status of that idea? Because as you mentioned, the 1 kilogram dose was rather arbitrarily introduced when it was sort of 40, 50 years ago.
It's been years since I've heard people talking about this. 3 or 4 years ago, I know Yves Henon had organized a group of people to bring this up and see how we might best address making a change, either to bump it up to 1.5 kilograms or to eliminate it altogether. I don't know what progress has been made on that, or if anyone's still thinking about doing that.
There was a related topic of energy levels on electron beam as well, wasn't there? 5 MeV, 7.5 MeV, I think, was another of the topical questions. I'm not quite sure where that's up to. Presumably,
right. Maybe someone in the audience knows more about that. I really haven't followed it for the last few years.
We'll look into it and come back. We'll try and find out what the current state of play is. There's another post here. Are there any studies using irradiation to control disease, similar to thermotherapy?
Interesting. Are we? I'm assuming they're talking about diseases on the surface of fruits. Or are they thinking about something altogether different?
Exlia flavacin dori. Does that make sense? So
I'm
controlling
not sure.
disease similar to thermotherapy.
I'm not sure how to answer that question. Irradiation at the doses we use to control insects may cause something on the order of a 1 log reduction in surface pathogens on fruit, but that this phytosanitary irradiation is strictly for controlling insects, mites, other quarantine pests, not diseases on fruit.
Yeah. I mean, it's actually fascinating, isn't it? That often people group together food and phytosanitary. I often liken it to, sales and marketing. They're very different concepts. They're done for very different reasons. And, food safety and food security, as opposed to border control, phytosanitary irradiation, are very different needs and applications.
Yep, good point.
Let me just see if there's any more questions. I can't see any here. I mean, Peter, it sounds to me as if there's a lot more research that can be undertaken in all sorts of different groups of pests and as the world evolves. And it also sounds as if the biggest challenge is actually getting consensus views as ever from the regulatory community to recognise, particularly as you say, moving from the order to the family level, if you're going to expand the application of comprehensive generic doses. I think it sounds as if those are two of the big asks as we go forward.
Great, good summary, Paul. Thanks. Thanks very much for inviting me to speak. I enjoyed it, and please, anyone who has more questions, feel free to call me or contact me.
Well, let me just go through, and thank you, Peter. There's a few sort of closing remarks I'd like to just make. One is that there will be a phytosanitary session at the International Meeting on Radiation Processing that will be held in Costa Rica in November of this year. There is a phytosanitary strand there. IMRAP has a dedicated website, imrap-iaa.com. The program is now up there. The registration is open, so please visit that, and hopefully we'll meet some of you there. We're also looking into having a phytosanitary workshop on the day before IMRAP commences, so on Sunday, the 3rd of November. And that will be very much a sort of mentoring exercise, a very interactive, engaged workshop to try and help people who are looking to make investments in infrastructure to support phytosanitary irradiation. So that is actively being worked on, and there will be some communications coming out in the next few days. That may include a questionnaire. It will include some more details about what we're trying to achieve. So I would encourage you to register at the PsIP website, both as a member of that. There's no cost associated to it, but also tick the box to say that you sign up to the to newsletters and so forth, so we can include you when we're communicating some of this to the group and to the community. And the final point is that in the opening remarks, I referenced a book that Peter had co-authored in 2017, Irradiation for Quality Improvement, Microbial Safety and Phytosanitation of Fresh Produce. You can, of course, get that by searching on the internet, and it's available to purchase and download. If you'd like an electronic copy, Peter has made that available. We will put that in the information hub, the members area of the PsIP website, so you can download that. So please take a look at that, and we will upload this presentation onto the PsIP website shortly. So it only leaves me to thank Peter for his excellent presentation and thank everyone for joining us. And we will, because of the time difference, we will be able, because we've recorded it, to reach a wider audience. So with that, thank you once again, Peter. Thank you everyone for being with us, and goodbye. Thank you for your time.